Honorable Dr. Biplab Kumar De sir has already joined with us. So I would like to request our respected Dean sir to welcome our guest speaker. Please, sir.
Thank you, Paul. Very good morning to one and all present out here. Um, in the very beginning, I would like to take the opportunity to welcome our today's speaker, Dr. Supratim Resa, who has been known to me, and I am known to him since a long time. And we are really fortunate enough to have him today with us. I'm sure the upcoming uh, lecture session given by Dr. Rai would be very much informative, and students, researchers, and faculty members are going to take the leaf from all those discussions and the contents that would be presented by Dr. Rai. And I would expect that more number of participants by now should have joined, but I'm sure that they would be joining down the line once the lecture would be started. So this is appropriate effort that we have taken. Of course, now the offline and online blending has been adopted worldwide. Still, we have started this FDP with the online mode. Of course, we will be shifting to the offline mode eventually. But during this online mode also, the faculty members and the researchers will not be deprived of dissemination of the knowledge and the feeding information, recent advancements in the teaching, learning, research, and academics. So with this, I will once again welcome Dr. Supratim Rai sir. Thank you for joining with us. Over to Mr. Gogoi.
Thank you so much, sir, for your kind words. Before starting the technical part, I would like to provide a brief outline of today's speaker, Professor Dr. Supratim Rai. Dr. Supratim Rai is currently working as professor in the Department of Pharmaceutical Science, Assam University, Silchar. At present, he is also the dean of the school. He did his B.Pharm, M.Pharm, and PhD from Jadavpur University. Dr. Rai has more than 22 years of teaching experience, and his area of interest for research are drug-lipid interaction, QSAR, QSPR, QSTR, and molecular docking. Dr. Rai has published more than 60 research papers in various national and international journals. He had authored several books and book chapters. Now, I would like to take this opportunity to invite Professor Dr. Supratim Rai sir to please deliver the lecture based on the topic Utility of In-House Softwares in QSAR with reference to Electrotopological State Atom Index. Please welcome, sir.
Okay, thank you, Dr. Gogoi and Professor Biplab De for inviting me. I also extend the thanks Dr. Minmoy Basak, who initially contacted me, and Dr. Shanto Mondol for all the communications. Now, when I see the theme of the FDP, so I told that directly I cannot speak on these lines. So I can say some, or I can share my views regarding some work which I have done little early. And in this COVID period, there is a shifting of work from offline to online, and there is a much more work is going on onto this in-house because the laboratory also sometimes closed. So based on this, I prepare a slides I will share. This is my earlier work of one of my students. Along with this, we have done it. So let's start this. It is clear?
Yes, sir, it is clear.
Okay. So I have prepared the topic in this way that some software myself along with my computer colleagues in my earlier organizations, we developed say, some in-house software for electrotopological state atom index calculation parts. So with that software along with other software, we have done some work. So my today's discussions will confined to this. I will discuss a little bit about the backgrounds, then what is electrotopological state atom index or E-states, the software which we developed, that is the name is given is SR-ETSA, and some examples or some work what we have done with these softwares. So if we go by definition, then the drug designs. So in a simple words, that means the planning and concept by which a candidate can be selected as a therapeutic agents will carry the label drug designs. Now, rational drug designs and QSAR are two inseparable terms. It is better to say they are the two sides of the coins. If anyone insists for the demarcation, it may be said that QSAR is the way to RDD. Now, people have tried many things to represent the structures in terms of numerics, in terms of number. So all of these ways, there are many ways or many techniques are there. So all of them, electronic and the topological factor is very, very important. Now, electronic factors includes the polarity, charge, energy level, and the topological factors mainly includes the arrangements of atoms, then skeletons, then size, shape, etc. And the relationship between the various bonded or non-bonded parts of the molecules. So for the developments of the non-empirical structure descriptor for QSAR, attention was given for representing the whole molecules. These indices mainly are based on atoms, bond type of bonds, usually have presence into the molecules. This work actually initially started by the Randick. Then later on it was carried out by Kier and Hall, that is the connectivity index. So according to them little bit, the structural specificity of a drug molecules is exhibited at an atomic or fragment level instead of the whole molecule. Because in the drug receptors interactions, a part of the molecules, that is the pharmacophore, which play important role other than the other segments. So based on these concepts, there are basic many informations about the topology, about the electronic factors, about the atom levels, that is the count of atoms, bonds, path bonds, etc. All are applied for the summing up to develop the numerical form of the structures. That's why the QSAR studies on the atom level or fragment level are justified in this present context. So what is electrotopological state atom index? Electrotopological state atom index, it is developed by Kier and Hall. It is an atom level descriptor and it gives the information both electronic character and the topological environments of each skeletal atom in the molecule. Now, in a molecule, each atom exists in a field composed of every other atom in the molecule. Now, those fields defining the uniqueness of any atoms. Because the atoms are surrounded, the type of atoms present in a molecule depends upon and determine the environments. For example, in this case, there are two similar methyl groups are present. These two similar methyl groups are chemically equivalent because the impacts of the field within the molecules are identical. But the third one is the same methyl groups, but its environments is little bit differ from the two. So this actually it is this impact of each atoms on a given molecule which provide the informations in this field. This leads to the state atom index with the molecular skeleton. Now, how this electrotopological state atom index is developed, it is like this. Kier and Hall, when they started to work with this, that is the electronic and topological parameters, so they first try to determine what is the internal or the intrinsic characteristics of the atoms. Intrinsic characteristics of the atoms and the effect of the surrounding atoms. Now, it must include the both topological and the electronic factors. Now, electronic factors, only the non-sigma electron, that is pi, and the lone pair of electrons are considered particularly because they are reactives and closely associated. Later on, they take the contribution of the sigma electron also during the development of the process. So they find that the pi electrons and the lone pair of the electrons, they are considered, they are reactives and closely associated with the stronger long-range interactions is there with their valence state electronegativity of the second quantum level covalent bonds. So non-sigma electron counts they have done, that is delta V minus delta, where delta V is the ZV minus H. ZV is the number of the valence electron and H is the number of the bonded hydrogen atom. And in case of delta, where they count the sigma electron also, that is the count of sigma electrons orbital, and the H is the number of the hydrogen atom. So that's in this way they considered both pi lone pair along with the sigma also. Now the topological attributes in the intrinsic value is the atom relative location within the molecule, that is the relative degree of mantle atom or buried atom status. So the count of the nearest neighbors in the skeleton is used. Now delta is used as a measure of degree of mantle of atom status. For terminal atom, where delta is equal to 1, atoms are on the surface or mantle of the molecule, whereas the atoms, if it is delta is equal to 2 or 3, then the effect is difference. So initially what they thought, they take delta V minus delta by delta to consider as the intrinsic quantity of the interest. But they found that the hybrids of an atom, particularly carbons, having the same values. So these groups have different chemical role in biological activity. So to solve these problems, they add a constant to the numerator, that is delta V minus delta plus 1 to resolve this problem. So finally, the equation was simplified by adding 1 to the whole expressions, and the new equation was adapted to express the intrinsic values of the atoms. So intrinsic factors, they have calculated by this formula, and the different structural skeletal of the hybrid groups is different. So it is found that the terminal acetylene carbon possesses the highest value of 4, and the quaternary carbon has the lowest value of 1.25. So these values agree with the notions of the electronegativity also. So they develop the intrinsic factor in this way. Now in series, particularly with when fluorine, hydroxyl group, NH2CH2, and other groups are there, then there is a constant value. To reflect these differences, the principal quantum number is modified. So what they did, to solve this problem, again they modified it. So they add the second quantum level and the formula is modified in this way. So ultimately for electronegative groups and hybrid, that is different state of carbon atoms, finally the intrinsic contributions of a particular atom is determined by this formula, where all the options or all the contributions were considered by the Karen Hall. Then what they did, the contributions of the nearest atoms, that is the distance. So the field effect on each atom, the intrinsic values then are used to quantify the influence of one atom to other. The effects of the field of an atom is a summation of the effect of all atom pairs. Now the field has a perturbing effects on the intrinsic atom value. This perturbation is assumed to be some function of the difference of intrinsic values of two atoms, say II and Ij. So the perturbation factors can be calculated from this value. Now, the influence of a particular atoms in a molecule, there are various numbers of atoms may be present, and their distance of a particular atom also varies. So the shorter distance atom contributions and the contributions of the longer atoms which are present in a long distance, so this all should be considered by considering their distance, that is called the graph separations. So finally, the perturbation factor, that is the effects of the other atoms along with this distance, is calculated by this formula. So electrotopological atom, state atom, is given by the formula, that is the intrinsic factor plus the perturbation factor, that is the effects of the other atoms. Now the next is how to develop a software based on this. So the principle we learn, now we try to develop some software. So myself along with my colleagues at my earlier organizations, we developed this. So this is the software. SR, we have given the names, and the computer, my colleague, the Ratan Bishas, and myself, we developed this. So when you open this software, so the screen will be like this. There will be 4 options: file, tool, window, and help. Suppose this is a simple molecules. Simple molecule is there. So initial step will be you make an arbitrary numbering of the molecules. Arbitrary numbers. By keeping, suppose in a series there are 20, 30, 40 molecules is there. By keeping the common atoms, do the same numbering. But the substituents may have the different numbers. So it's a simple molecule we have started. So we have numbered this like this: 1, 2, 3, 4, 5, 6, the benzene nucleus, and the substituent 7, 8, 9 is the substituent nucleus. Clear? Then in the files we have to load the new distance matrix. So when you load it, suppose in this case the number of atoms including all are 9. So when you click this, that the new distance matrix, it will ask that how many columns will be required, including the intrinsic factors. So we have seen that here in this case there are 9 atoms are there. So 9 atom plus 1 intrinsic factor, it is 10. So when you create it, it gives a distance matrix with the intrinsic factor, it will give a column. Now the purpose is this: we have to, suppose there are 9 atoms, we have to find out the distance of each atom from another atom. For example, say atom number 1. So atom number 1 to 1, the distance is 1. 1 to 2 is 2. 1 to 3 is 3, like this. Similarly, 1 to 7 is 2. 1 to 6 is 2. 1 to 5 is 3, like this. Say 1 to 8 is 1, 2, 3, 4, like this. So in the next, when you go it, the diagonal values, cell values. If you see 1 to 1 is 1, similarly 2 to 2 is 1. So when you click this that is inside diagonal cell values, it will come all 1. Clear? That means 1 to 1, the distance is 1. 2 to 2, the distance is 2. 3 to 3, distance is 1. 4 to 4, distance is 1. 5 to 5, distance is 1. 6 to 6, distance is 1. 7 to 7, distance is 1. 8 to 8, distance is 1. And 9 to 9, distance is 1. Now the individual distance between 1 to 2, how much? 1 to 3, how much? 1 to 4, how much? That we have put like this. Suppose here, for example, the atom number 1, the first row. So 1 to 1 is 1, 1 to 2 is 2, like this, 1 to 3, 3, like this. And the intrinsic value of 1, 2, 3, we have added into the first column from the reference tables. Clear? Then you have to click the generate the results. When you generate the results will be coming. The E state value will be at the last column. Whether the program is or our work is correct or not, that can be checked by the delta I value. It should be 0. It should be 0, near to 0. Now in the second column, you see the summation is near to 0. So when we go to the save, for the saving of these results, save the results, it will save in the Excel mode. When you save in the Excel mode, it is easy to take it or easy to copy it rather than to write it and type it. Okay? So this is an example for this. Now, the utility of this state. A QSAR study generally involves 2 steps. First, the descriptors are generated, which encode the structural informations. And the second step is that the QSAR should be, and the second one, we have to perform the regression to determine the relationship between the various parts or various structural information with the biological activity. The descriptors used in the QSAR should be meaningful and it can be easily interpreted in physical terms. The descriptor actually should provide information into the mechanisms, and the statistical regression method much more correlate changes in the structure with the changes in the biological activity. Now to do the work with this, our in-house developed very small software, it is very small. So what we did, we take some hydroxyphenyl urea derivatives reported by Yusuf et al. Having free radical scavenging activities. And we try to develop the model using states and some physicochemical parameter, mainly 2 parameters we have taken, the log P and the molar retractivity. The purpose of this modeling is to identify the features of the molecules contributing the antioxidant activity and the designing of the new molecule in this class. So this is the structures. This is the structure. There are 36 compounds are there. And there are structural variations, 1, 2, 3 positions are there. 1, 2, 3, 4 positions are there along with the N. And the log P is given. So what we did at first, the activity, that is the free radical scavenging activity data, that we convert to log 1 by this activity to determine as a response variables. Then along with this, the partition coefficients and the MRs and the electrotopological state parameters are calculated. The log P partition coefficients and the molar refractivity of the compounds are calculated from the ChemDraw softwares using Ghosh and Krippen's modules. E-state parameters were calculated by the in-house software SRXA developed by the partly by the presenter and the standardized on the known data sets. The program uses as input only the connection table in a specific format along with the intrinsic values of different atoms. And the output file thus we obtains the biological activity are introduced and perform the regression analysis. So this is the compound type and numbering. So I have made the common numbering of all these things. I kept the hydroxyl group at position number 2 and including its number also in the common number parameters. So after a calculation of the state parameters, so what we did, we first divide the data sets. Now there are several logic is there, several OS is there, and in our case we standardize or we found that the 75-25 ratios by K-means clustering we have applied it. Now there are other techniques also for the division of the data sets, and these actually determines on the prediction ability of the models. So QSR models were developed using the training set compounds that and optimized by the Q-square values, and with these models the external that is the test sets were predicted. So two statistical regressions were performed, one is stepwise multiple linear and another is the GFA genetic function approximations. So stepwise and MLR were performed by the Minitab software and the GFA was performed by the Series 2 version software of QSR plus environments. So what we did, this is the division of the compounds in various clusters. There are 5 clusters are there. This is the numbering of the compounds. And the statistical models what we develops, that is the square correlation coefficient, variance ratio, then the for GFA, it is the lack of fit was taken as a objective function to select an equations. And the generated QSR equations were validated by the leave one out cross validations and the trace values. And for external predictions, we have the R-square predictions and the 2 matrices developed by my guide, who is the next speaker. So for the stepwise regressions, now the stepping criteria actually we have kept F is equal to 4, that is based on the variance ratio. F is equal to 4 to F is equal to 3.9. There is a logic behind this. So we try to at 95% level. So we try to keep this. We have tried with other variance ratio also within the limit, but this F4 to 3.9 gives a better one, and another logic also there. So after developments of the model, this is the stepwise regression model. So we found that the S6, that is the position number 6, 6 that is in the structure, then S4, and then S7 has negative contributions, have the negative contributions towards the activity, whereas log P and S5 have the positive contributions towards the activity. Now, when we explain it, for the explanation, we found that for position number 6 particularly, the presence of CH3 substituents at position number 6 for compound number 33 to 36 having lower activity. But the presence of the methoxy substituents at position decreases the state value at position 6. Then when you come to the S7, it is also the negative contributions. We found that the higher range we have negative contributions towards this activity. Then presence of methyl groups at R double dash position increases the value of the S5. S5 have positive contributions. And the partition coefficient has a positive, that is particularly N octanol water partition coefficients have higher, have positive regression values. And number of the methylenic fragments into the side chain also have contributed towards the activity. Now, in this case, the developments or the statistical significance, particularly we have found the internal prediction is good, quite good, 0.953. Praise value also less. And the R square prediction, that is the prediction with the test set, also good, 0.916. And the RM square, that is, and the for the test, for the training, and the overall, all are with the above the stipulated limit, and it is a statistically significant model. Now, in case of GFA, we have used two techniques, mainly one with linear, another with the linear and spline. The linear and spline has a better interpretation capability due to its range of the values. Now, in this case, by GFA, in this case, we have performed 5,000 iterations. Now, the Q square value also high, the R square prediction also high, and the RM square value for all these are above the stipulated limit. Now, in this case, the S7 negative, S8 negative, S1 negative, only partition coefficient have the positive contributions. Now, S8 particularly, the parameter indicates the importance of the amino nitrogen atom of the hydroxyphenyl urea for the free radical scavenging activity. And the S1 indicates the E state atom is identical for the activities, and the E state value of atom number 1 is influenced by the nature of the substituent which is attached. Then when you go for the spline, GFA with the spline, here you can see along with this there is a range is given, either positives or negative is there. So this will be helpful for understanding that how much limit you can go. Now, in this case, the positive coefficients of S7 along with these values indicate the E state index of atom number 7 is conductive for binding affinity when the value is less than this. So, from this, the advantage of spline is that you can also quantify that how much limit you can able to go. And the Q square also is much more higher. Q square, R square predictions, all are very, very good. And I think this is one of the best model for this. So, what is the conclusions? All the developed models particularly indicate the important contributions of the phenolic hydroxyl groups to the free radical scavenging activities. Then hydrophobicity, particularly the partition coefficients, is very important for the free radical scavenging activities. Then the number of methylene fragments in the side chain and the nature of the substituents are triple dash, all influences on the hydrophobicity value of the compounds. Then we found that the compounds without any substitution at R dash, R double dash show better activity. Specifically, it was also observed that CH3, OC2H5 also respectively decreases the activity. Then the results indicate the importance of the urea moiety and the piperazine nucleus in the side chain for the free radical activity. And one of the interesting findings is that in all the models, the differences between R square and Q square is not very high. It is less than 0.3. That's also one of the good thing of these models. And based on the RM square overall criteria, GFA spline was the best models. Now, finally, we conclude that apart from the presence of the phenolic hydroxyl moiety, hydrophobicity, appropriate substitutions, all these are important for the free radical scavenging activity. Now, in this work, there are many work is pending. Many work is pending, or we have not done it also. That we will do it. Applicability domains, then the MSc, that to to see the robustness of the model much more. That and only stepwise regression and the GFA was done. But along with this, the other statistical techniques also not applied. Particularly the PLS, partial least squares. This is one of the best regression techniques that we have not applied here. So these are the future things we will do. Now this model was developed with the E-state and the two physicochemical parameters. We have to develop these models with lots of descriptors freely available. Lots of online descriptors are available with Paddle and others. That can be done. And that can be compared also with the E-state parameters. But we found that one of the observations and the other works also, if the number of compounds is not so much, suppose here it is 36. If the number of compound is more or less 30, 40 or like this, and the structural variation is not too much, E-state has a good interpretation capability. Ultimately, you have to interpret it. So with this, I finished. Any questions, if I able to give answers?
Okay, if anyone has any question, ask him. If anyone has questions, please come forward and ask him.
Sir, good morning, sir. Professor Supratim Ray, sir.
Good morning.
Sir, the software what you have developed, you were saying that it is used for identification and like numbering everything. So is it like only for doing 3D QSAR? This software?
No. It is not for 3D QSAR.
Sir,
it is only 2D, two-dimensional, only 2D.
Only two-dimensional. Okay. A two-dimensional QSAR.
Because E-state only 2D, 3D QSAR is docking and other parts.
Yes, sir.
But it is not related. It is only two-dimensional QSAR. 2D
so
QSAR.
The parameters, whatever the parameters like steric, electronic, everything can we know by using this software, sir?
No. This software is for electrotopological state atom index. Only E-state.
Okay, only for the single individual atoms.
Only for the E-state parameters.
Only
Not for any. There are 4,000, 5,000 descriptors. Now, if you go for the ALVA-based software, Paddle, if you give a single molecule, they will give you 3,000, 4,000 descriptors. It is only single descriptor E-state.
Sir, is it applicable for larger molecules like proteins and amino acids, or only
No,
molecules?
No, not larger molecules. No larger molecules.
Yes, sir. Thank you, sir.
Okay.
Is there anyone who wants to ask the questions?
Okay, so I extend my sincere thanks to Professor Biplab Dey, Dr.
Okay,
Bosat,
sir.
for giving me the opportunity. Now, if you allow us, I have some important phone call is coming.
Okay,
I
sir.
can
Thank
conclude
you
here.
very much for your magnificent presentation, sir, and enlightening us with your knowledge and experience, sir. Thank you very much, sir, from our
Okay.
department. Sir.
Thank you. Thank you.
And for all the participants, feedback form will share in the chat box. Kindly fill the form before leaving. And thank you for valuable presence. Stay safe and have a good day. All of you, please fill up the feedback form and you may leave now.